It's Dr. Peter Loskill. He's professor for organ-on-chip research at the Eberhard Karls University in Tübingen in Germany and the Natural and Medical Science Institute, NMI, and he's vice chair of the European Organ-on-a-Chip Society, the EUROoCS. He received his PhD in physics from Saarland University. He's currently heading the Microorgano Lab and the 3R Center in Tübingen for in vitro models and alternatives. He was named in technology reviews as one of the innovators under 35 Germans. Companies and awarded by the Fraunhofer Track Starting Award. So his talk today is about EUROoCS and encouraging and develop organ-on-a-chip research in Europe. Stage is yours, Peter.
All right, good morning from Tübingen, Germany again. I'm happy to talk today again about an important topic, not our research, but about how we're trying to bring all the stakeholders together in organ-on-a-chip research here in Europe and also in beyond. And to give you a little bit of an overview of how we got there and how we set up the European Organ-on-a-Chip Society and what the current state is, I'm going to take you to a couple of slides. I will not need the entire 30 minutes. I might be able to help you get back on schedule, actually. So let's get started. As I mentioned, I want to first set the stage a little bit on how it all began for EUROoCS, and then talk about our mission and vision and then the current state. How it all began. It basically began with an EU project called ORCID, and this project, short for Organ-on-a-Chip in Development, was a Fed Open CSA, which is not a typical EU research project. It's more of a road mapping project. So there was no research conducted in a classical way, but the idea was really to create a roadmap for organ-on-a-chip technology and to build a network of academia, research institutes, industry, regulatory bodies, and patient organizations in Europe, and to raise awareness by disseminating and communicating information about the technology, the current state, and the potentials and the limitations. The website should still be up, but this was the starting point. And what we did during ORCID was a number of different, let's say, studies or different work packages. And we started with just looking at the bibliometric approach, looking at how has the publication and patenting process been over the last couple of years. And so this is in 2017, because that's when the ORCID project started. And it was also the reason why the ORCID project was implemented. We saw a sharp rise in publications and patents over the last 10 years with respect to organ-on-a-chip technology. And this was very interestingly visible in Europe and in America and in Asia at the same time with a very similar rate. So you could really see that from the 2000s on, the technology was taking off. There was also a large patenting drive. So there's a huge number of patents now out there, and some visible, some not yet visible. And so based on this background, we started the ORCID project, and we started it in two ways. We did first a bibliometric approach and a market study, and a lot of interviews with experts, a lot of workshops with stakeholders, and really the idea was to kind of map the current state and the roadmap. If you're interested, there's a lot of publications out of that. I'm not going to cover all of that, but there were a couple of major outcomes. One was, for example, we came up with an organ-on-a-chip value chain where we tried to really plot all of the different disciplines, all of the different components that go into an organ-on-a-chip system, and showing really the interdisciplinarity of it. Another outcome was to really look into, because there was a lot of talk about the potential of organ-on-a-chip systems, and still today you hear a lot about the potential of organ-on-a-chip, how it can be a paradigm shift, how it can be change the way of how we do research. However, you have to be very careful with discriminating what's the hype and what is actual real potential. And so we tried to get a little bit of numbers around it, and we did a budget impact analysis. Because at this point, and even today, it's really hard to see how much could the organ-on-chip technology actually impact pharmaceutical R&D. So what we did, it was a very talented PhD student, Nora Franzen from Eisenman, and she did a huge interview series with experts from mostly end users, pharma, biotechnology, but also academic end users and some organ-on-chip developers and regulators. And she had a huge questionnaire and then combined these results by linear opinion pooling and weighted that for experience. And what she came up with was that in the most optimistic cohort, you could see that there's a R&D cost reduction of almost 26 to 35%. In the average cohort, the hope or the kind of belief was that we will have an R&D cost reduction of 10 to 26%. And in the more pessimistic cohort, you could see an estimation of a 10% reduction or even a 5% increase in R&D costs. And what does that mean in numbers? It's always very tough to get total numbers of R&D of pharmaceutical or drug development. There are a number of publications out there from 2016 and 2017 from Prasad and Dimasi, and they have already very different numbers. And the actual numbers might be, again, different depending on which big pharma you look at. But just to have an idea what that would mean, the idea could be, these numbers would mean that we are talking about savings in the range of 100 to 200 million dollar per drug in the case if we believe the numbers of Prasad. If we look at the numbers from Dimasi, it would even be from 200 to maybe 900 million dollar per drug. So these are the total numbers, and behind that there was a lot of analysis and major cost driver that were identified in that study was not per se the cost of running an assay or the cost of running through the pipeline, but rather an increase in success rates. So already at that point, the experts did not believe that per se organ-on-chip studies or organ-on-chip experimentation are going to be cheaper than traditional, let's say, animal or other in vitro methods, but that the predictivity will lead to a significant reduction of drug prices. All right, so this was one of the, let's say, more the economic outcomes of ORCID. And at the same time, there were a number of other stakeholder workshops and efforts in mapping similar things. And one is, for example, was a stakeholder workshop run by a think tank in Berlin, a transatlantic think tank. And there was also focus on pharmaceutical R&D and checked where could these models be implemented in the drug development pipeline. And basically what came out there was that at any stage of the current drug development pipeline, both in terms of efficacy or safety, MPS could be used for specific applications. The question, and this is a question you hear still also today, is that actually happening? And because this is something very, very difficult to assess, because obviously a lot of, especially the most successful things running in a pharmaceutical company are not per se being published or it's not publicly available. This think tank tried to get a little bit of an assessment by using all of the stakeholders that were in the think tank from the pharmaceutical industry to see what they could disclose. And you could see that there are, at this point, this was 2020 published, but the stakeholder workshop was 1 year ago. 1 year before, you could see that there are already a number of different models in different pharmaceutical companies from different suppliers being used in different types of drug development, in different types of applications and at different points during the drug development phase. So you can see this is very, very likely only a small subset of what was disclosed, but that means that there is an increasing adoption by pharmaceutical companies. Last year we did actually a fun brainstorming experiment, which basically built up on that and other think tanks and workshops before, which always try to look where can they fit the MPS and organ-on-chip technology in a classical pharmaceutical drug development pipeline. So what we did is what kind of think a little bit different. Okay, if we would now forget how pharma R&D is done, and the nice thing is you can, on paper, in a perspective, you can do just very bold ideas. And so together with Ryan Hardwick and Adrian Rode, we try to just brainstorm. If we would have a look at all the models we have available now, how could we design a very different type of pharmaceutical R&D? And this was really, this was what we came up with a fully human-based approach where we have high throughput stem cell assays with a low complexity but high standardization aspect, and then low throughput, high complexity multi-organ chips. And it is all the way we can go with the same type of cells and standardized cell sources. And this could be embedded in big in silico frameworks and then supplemented with animal studies wherever they need it. For example, in the beginning, an in vivo PK study to define which organ is of major interest, and in the end, a pivotal in vivo tox study. And this then could really be combined with biobanks and databases and would lead to a very different way of a more human-centric pharmaceutical R&D, or even in general biomedical research. All right, so these were a couple of let's say, studies that were going on during and after ORCID. But what we realized in ORCID is that we have all of that potential, but there's also a huge amount of unmet needs and challenges. I'm not going to go in detail through them. In the last 3 years, we might even have resolved some of them already. But there is a lot of different challenges and needs at different levels. And one of the issues we identified is also what this gap between what academic developers, academic researchers produce and what industry requires to really adopt it in a standard workflow. And the problem is that academics typically develop a model, then focus on a proof of concept, mostly only in the developer's lab, sometimes in collaboration in other labs, and more focus on a functional validation and mechanistic applications, and typically use some compounds as validation compounds. However, or as reference compound, let's call it like that. What industry would really need is large reference data sets, a high technology readiness level, and automated handling and intra-laboratory reproducibility, and also validated systems. The issue is here that if you have developed a model, have introduced it, have published it, it's very hard for an academic to get funding to just test lots of compounds, where you already know what the effect should be. And it's also very hard to publish that. So this is not something a PhD student or a postdoc could do. Because of basically the way academia is structured, and also because of missing funding mechanisms. So we had a couple of ideas how we could bridge this. And one is obviously funding. That's always in academia, funding solves everything. Not everything, but it's the hope. But there's other aspects, such as involvement of regulators at very early stages. Then what was already implemented in the US, the tissue chip testing centers. But then also 3 aspects which we identified as where something we can really work on. And that's on the one hand the standardization, on the other hand tailored training programs, and on the other hand bringing all of the stakeholders together. And this bridging function, this is what the EUROoCS is supposed to be. Before I come to EUROoCS, very briefly on the training, we also did a lot of work in terms of what is needed in terms of training and what kind of training programs should be set up within ORCID. And we did this again in a huge questionnaire where we approach stakeholders such as developers, end users, but also regulators, and try to figure out what are the most important aspects to be trained, for whom, and at what stage. And so we have a lot of, let's say, and did a lot of analysis. This is published in the ORCID report, but will also be published in a manuscript hopefully soon. So if you're interested in setting up a training program, I can strongly recommend this as a resource. We have tried to implement that also ourselves, for example, in an ITN EUROoCS training program, but also in locally here we are running summer schools at the 3R Center. All right. So this was one of the main outcomes in terms of training. But the major outcome of ORCID was really to come up with this roadmap. And this roadmap is now a little bit here in the schema here. But the key really was that we need somehow an instance which really fosters and facilitates this roadmap. We can really go from initial development to applications in lots of different areas, not only pharma, but also in food, cosmetics, environmental, but also hospitals, clinic, and very importantly also academia. So to foster this roadmap and to facilitate the implementation of this roadmap, we then founded EUROoCS. And EUROoCS was founded as an independent non-for-profit organization, and the primary goal was to encourage and develop organ-on-chip research. And this was the hope to be, we were able to implement, or to do that by implementation of this roadmap. It was launched in 2018 by Christine Marmory, Janne van den Eyden, Albert Vandenberg, and myself. And that was basically the starting point. And the mission and the vision EUROoCS has was something we now we went through a strategy process in the last 1.5 years to really sharpen the mission and the vision, and to establish new ways of really implementing this roadmap. So the mission and the vision of EUROoCS are, and because the mission is really to enable organ-on-chip technology to facilitate life science research, promote human health, and improve quality of life for all patients. So this is the mission. This is what EUROoCS is working towards. And our vision of how we actually will do that is really to facilitate organ-on-chip scientists, industry, government, regulators, and general public in realizing sustainable progress in biological and medical research by education, communication, funding, and matchmaking. And here the beneficiaries are pharmaceutical, cosmetics, chemical, but also agri-food industry, which can have an impact directly on consumer products. And so we try to have EUROoCS as a catalyst for change around the experimental use of animals. So the 3R aspect is also very important. And we want to facilitate that organ-on-chip technology can become a standardized and reproducible way, so that promoting adoption by regulators, academia, industry, and the general public. So this is the general vision of EUROoCS. And how are we doing that? So what is the current state of the society? So we have today more than 500 members from all over Europe, but also from the rest of the world. So we are open to anyone, and we actually have, I think, almost from every continent members. The society is governed now by a board of 12 elected members, which will be increased even further next year, where we have election with, I think, the last 2 missing board members. So it will be so then kind of it's a periodic way of elected members. It is supported by a regulatory advisory board and an industrial advisory board, and this really goes back already to these findings that we need to have all of the stakeholders involved already at this early time point. So even when an academic starts a new project, thinking about a new chip, it's important to already at that point be aware of regulatory requirements and be able to have or have the knowledge about how this could be used later on. We have an alliance with the ISSTR, the International Society for Stem Cell Research, and a home journal, Stem Cell Report. So every EUROoCS member gets a cheaper publication fee there, and we have a number of EUROoCS members on the board to actually have a good editor base for editor and reviewer base for organ-on-a-chip applications. It's mostly focused on organ-on-chip systems that use and implement, combine it with stem cell technologies. We're also working in another home journal, which would be more of the technological aspects. And in general, what it already does today is the current state, it really fosters interdisciplinary and intersectoral collaboration. As I mentioned, we have 3 boards, very briefly here. These are the 3 boards. So we have the society board, which I'm chairing, and Andries van der Meer is the vice chair. Christine Mammery is the past chair, and we have Jannie van den Eyden as a secretary treasurer, and then Albert Vandenberg as a member, Dries Bracken, Erika Giovanni, Thorsten Meyer, and Martin Rasch, and Marco Rasponi. And we're already spanning both industry, non-university research institutes, and companies in the board, and also a broad number of countries. And then we have our 2 very important advisory board, with the Industrial Advisory Board, which is chaired by Pelin Kandaroglioglu, which includes I'm involved, Jochen Kühnel, Joshua Chiba, Barbara Birk, Lisa Benin-Koza, and Thomas Singer. And we have our Regulatory Advisory Board chaired by Maurice Whelan, including Susan Fitzpatrick, Susan Brentler-Schwab, Sonja Bacon, Natalie Delrue, Jose Tarazona, and Jacqueline van Engelen. And these boards play a major role in shaping what a society actually does. And how is the society implementing things? That's now one of the new things we set up in the last year. These are working groups. So we have now a number of working groups that have defined missions to really facilitate aspects of organ-on-chip research and adoption, and to organize society activities. These working groups have regular meetings and are in close coordination with the EUROoCS boards and the advisory boards. And they are open for participation of all EUROoCS members. We, for example, have a working group on digital meetings, so there will be much more webinars, for example, to come, chaired by Christine Mammery and Marco Rasponi. Then we have a dissemination and outreach committee, which basically takes up a lot of things ORCID has started. Then we have a technology and science implementation board, a teaching and training board group, a diversity and inclusion group, and a fundraising. And also, then we're strongly involved with Sint Zenelec and the European Commission Joint Research Center, GRC, established a focus group on standardization of organ-on-chip system, and that's chaired by Andries van den Eyden. And maybe one of the most important accomplishment or things the society is doing is the annual conference. So when we started, there were lots of different smaller conferences on organ-on-chip in Europe. Now we merged all of them, and we are trying to have a low threshold and affordable, especially for all the career researchers, conference, which allows plenty of time for interaction and socializing. We started in 2019 in Graz, Austria, where we had about 200 participants. Then the next 2 years, we unfortunately had to go online, but we increased our participant number from 400 in 2020, 440 in 2021. And this year, finally, we could go back to a face-to-face meeting, which was in Grenoble. And we had about more than 460 participants from 25 countries, all over Europe again, but also from all over the world, actually. So it was a huge success. And next year, we will be organizing the MPS World Summit together with the CUT in the US in Berlin. So we will merge for next year the MPS World Summit series with the EUROoCS conference series and host the entire world, hopefully, in Berlin, Germany. So that's kind of one of the major events or major activities of EUROoCS. If you have more interest in EUROoCS, visit our website or follow us on Twitter, on LinkedIn. We also have a couple of information material, many stemming from the ORCID project, but also others, where a lot of the things I was talking about are elaborated on in more detail. For your kids and teenagers, maybe also interesting, we published a small paper in Frontiers for Young Minds explaining organ-on-a-chip technology. And with that, I'm at the end. I hope I could give you a little bit of overview what EUROoCS is doing and what the mission and the vision of EUROoCS is. I would be happy to answer questions.
Thank you, Peter. Well in time. We have time for questions.
Really enjoyed it. I didn't know about these organizations that you guys are doing from Europe and etc. Since India is becoming a major player in MPS systems, there should be a couple of people should be part of these memberships and advisory boards if possible, so that we can also bring back the knowledge of what is happening around the world into India and educate Indians to develop the right models and right systems, etc.
So the membership is open to everyone. So everyone can be, it's not limited to Europe at all. And so I encourage everyone to become a member, and also the board membership. It's anyone can become a board member. It's an election process by from all the members, and also there is no requirement that people are from Europe. So we would be happy to welcome members from India too, and to have an active involvement and active collaborations through that.
Thank you.
Hi, Dr. Loskill. Really nice talk. So I have one question, one very basic question. Is when we are trying to develop an organ-on chip, for example, placenta-on chip, so how do you set a proper control to compare it with the in vivo condition? Because sometimes the in vivo data is not available. So whatever data we get out of our placenta-on chip or any other organ-on chip, how do you know that if it is the right one, and how do you have a confidence on the chip we develop? Yeah.
This is not a simple question. This is it's let's call it the $1 million question, because I mean, and it's one of the aspects. For example, standardization work group focused on, but also there is the efforts from the IQ MPS consortium, which brings together industrial stakeholders. How do you actually compare? How do you validate what you see in your model is actually relevant? And I can't give you the answer, the general answer. As I mentioned, there's a lot of groups working on. One of the, and this is my personal opinion. The key thing is to try to find clinical comparisons, to not look into comparing it to what you see in an animal model or something, but really to see, can you implement clinical endpoints in your model that you could then directly compare to patient data. That's basically the perfect situation. In general, it is just the major recommendation I can give is to generate as much as possible data and to report this data transparently to see what your model can actually recapitulate in terms of mechanisms and what it cannot recapitulate. It will always be a model, so you always have to know what are the mechanisms that are actually captured. And the more you know about a model, the more you can also build confidence in what you can get out of the model. And this can be via case studies on let's say, mechanistic case studies. It can also be via testing of reference compound sets. And for a lot of different model systems, there are already lists of recommended reference compound sets. I don't think for placental models, for example, but especially for liver models, for example, or other models, the IQ MPS consortium has published a number of these reference compound lists.
Yeah, thank you.
We have time for one question. If not, then we will gain some time. Thank you, Peter, for this nice presentation.
Thank you.
And I will hand over now to Anushka.